calcium channels. So our work is on GABA A subunit and GABA A receptor. It allows the, it is basically a chloride transporter. It allows the passage of the chloride inside the cells. And it has a different subunits like 2 alpha, 2 beta, and 1 gamma subunits. Our work is basically on this subunit. And we have published this review in the Aging Research Review in recently, 2023. And we have found, this is some other article that we, it is published by some other researcher, not by our, but here we have recorded that in the Alzheimer disease, there is a low level of GABA and the enzyme and receptor and transporter. So overall, there is a reduction of the GABA inside the AD postmortem brain samples. So this is the basis of our hypothesis, and also our hypothesis is based on our some previous finding. So our main hypothesis is MIR-502-3p and the GABA in the AD. Is there any relationship? So in our previous study, we have found the overexpression of MIR-502-3p, and that causes the reduction of GABA receptor alpha 1. So this is our previous hypothesis and our findings. So we wanted to check, is there any relationship of MIR-502-3p that it causes the repression of the GABA receptor alpha 1? So for this is a brief material and method. So postmortem brain sample. Firstly, we have acquired the postmortem brain sample from AD patient with the different Bragg stages from 1 to 6, and unaffected control were obtained from the NIH Neurobiobank, USA. And we have used ST22 cells, that is the mouse primary hippocampal neuronal cells. MIR-502-3p agomir and antagomir were used for the microRNA overexpression and microRNA inhibition. And transfection we have done by the lipofectamine, and cell viability we have checked by Anaxin PI Nexlome kit. Then we have done the in silico analysis by RNA hybrid software for binding, RNA structure software for the structure prediction, and RNA full chain software for the entropy change. And moreover, we have done the biochemical assay that includes luciferase assay, QRT-PCR, Western blot, ELISA, in situ hybridization, immunostaining, confocal microscopy, patch clamp analysis to prove our hypothesis. So let us discuss all the results one by one. So this first slide shows our result for the GABA receptor protein level in AD postmortem brain sample. So firstly, when we acquired the AD postmortem brain sample and the unaffected control brain sample, we prepared the protein. And AD brain sample we have taken, selected on the basis of Bragg stages with the increasing of the Bragg stages. So those are the control brain the protein profile from the unaffected control postmortem brain samples of the age and sex matched, and then 5 patient sample from the different Bragg stages, that is Bragg stage 2, 3, 4, 5, 6. So we found, as compared to control, there is a reduction of the GABA receptor alpha 1 with the enhancement of the Bragg stages of AD. We found there is a reduction of GABA receptor beta subunit also as compared to control. And also as compared to control, there is a reduction of the GABA receptor gamma and also V-glut. And beta-actin is used as a housekeeping gene. This is the densitometric analysis for this band. This shows there is a significant decrease in the GABA receptor alpha 1 with the increased Bragg stages. And in beta, there is also a reduction, but as compared to alpha 1, it's less. Also in alpha 1 and gamma 2 and V-glut, we found drastic decrease or significantly decrease in the GABA receptor alpha subunits. So then this slide shows our previous result that we published in the NPJ Genomic Medicine in 2022, where we found different microRNA level with the increasing of the Bragg stages. We found MIR, and we have selected MIR-502-3p that is continuously increasing with the increase of the AD Bragg stages. We have selected this microRNA because on the basis of in silico analysis, my mentor has found out that it inhibits the GABA synapse function. So we are interested in the GABA synapse function in the AD. So we have selected this microRNA. Then this slide shows the in silico analysis for MIR-502 binding to GABA receptor alpha-1. So firstly, we have predicted the GABA receptor alpha-1 and microRNA binding by in silico analysis. We find out for this structure is a minimum free energy change is -30. So it supports the stability of this structure. And we predicted the 2D structure of this binding. This is the GABA receptor alpha subunit, and it binds to the microRNA. It shows the stability and the binding of this microRNA to the GABA receptor alpha-1. And we also studied the entropy change. When the microRNA, that is from 26 to 30 subunits, and microRNA at 90 to 98 amino acid position or nucleotide position, they have a stable structure. That's why they have the minimum entropy change. So it supports the stability of this structure. Then manually we find out different binding site, MicroRNA-502 binding site on its target gene GABA receptor alpha-1. We find out there is a 1 9-mer binding site. There is 2 8-mer binding site, 6 7-mer binding site of this microRNA on a GABA receptor alpha-1, and we find out there is a 28 6-mer binding site for this MIR-502-3p on the GABA receptor alpha-1. So on the basis of this in silico analysis and our Western blot analysis, so we want to check the MIR-502 is targets the GABA receptor alpha-1 expression or not. So first result for the MIR-502 modulates GABA receptor alpha-1 we have checked. Firstly, we have checked the microRNA binding to its target gene, that is the GABA receptor alpha-1, by the luciferase activity assay. We have performed the luciferase activity assay, and we find out when the GABA receptor alpha-1 and the artificial microRNA, that is a MIR-502 agomir, when both are present, there is a significant reduction in the luciferase activity. So it supports that the GABA receptor alpha-1 is a target gene for this MIR-502-3p. Then we have used the HT22 cells. We transfect them with the agomir and anti-agomir. Then we have prepared the RNA and we checked the microRNA expression by RT-PCR. So we find out when we transfect the cells with the MIR-502 overexpression, we find the overexpression of microRNA by RT-PCR. And more interestingly, when we check the expression of the target gene, we find out GABA receptor alpha-1 4 gene is significantly reduced in the cells transfected with the MIR-502 overexpression, whereas its expression is inverse. Previously, GABA receptor alpha-1 microRNA level is in negative, so its target gene expression in where we transfect the cells with the inhibitor of the microRNA, so they have the maximum GABA receptor alpha-1 expression. Same result we have obtained when we prepare by the Western blot. When we prepare the protein from the transfected cells, we find out that there is a reduction of the GABA receptor alpha-1 in the cells transfected with the MIR-502-3p overexpression. This is the same densitometric analysis. We find out there is significant decrease in the MIR-502 in the GABA receptor alpha-1 with the MIR-502 transfection. And same result we have obtained with the ELISA. When we prepare the protein and with the ELISA, we find out there is significant reduction of the GABA receptor alpha-1 when transfected with the MIR-502-3p agomir. And these results we have recently published in Neural Regeneration Research. It has been accepted for publication. Then we wanted to check either that this microRNA is toxic to cell or not. So We have checked the cell viability by the annexin pH, CC, and CCK8 method. So firstly, we have performed the cell viability. We have used the same ST22 cells and transfect with the agomer for the overexpression of the microRNA and antagomer for the inhibition of the microRNA. And we checked the cell viability after 2 time frames, after 24 hours and 48 hours. So we find out the cell viability has been decreased when the cells were treated with the MIR502-3p. Like this is the cell viability percentage of the cell viability. Previously, we have 81% viable cells, then they decreased to 78%. And as compared to inhibitor, they have 82.9% cell viability. And if we talk about the apoptotic cells, we find out there is 3.5 apoptotic cells, and they increased to 5.37 with the when the cells were transfected with the MIR502 overexpression. So this is the same results in the column method of depicted results for this observation. And same result we have find out that MIR502 overexpression transfection is the toxic to cell, and it decrease the cell survival rate by CCK8 method. This result has been accepted for publication in Neural Regeneration Research. Then we have checked the microRNA expression inside the cells by microRNA in situ hybridization. We have the same group of cells like control, and for the microRNA expression, we have transfect with the agomer. For microRNA suppression or inhibition, we use antagomer. But here we have cultured the cells on the cover slip, and then we have done the transfection and then microRNA in situ hybridization. So here we have find out the results where we transfect the cells for the overexpression of the microRNA. And here we can see that the microRNA MIR502 signals, because only here we have transfected from microRNA overexpression. So now we are interested to see the role of this microRNA on its surface GABA receptor alpha 1. So we have done the immunofluorescence analysis. So same way we have cultured the cells on the cover slip, and then we transfect with the scramble control, agomer and antagomer. And we have checked for the DAPI, merged image, and the GABA receptor alpha 1. So we find out in control there is a good GABA receptor alpha 1 at the cell junction or the cell networking, we can see. And when we transfect the cells with the microRNA overexpression, this GABA receptor alpha 1 at the cell junction has been reduced as compared to control. But when the cells were transfected with the inhibitor, there is an increase in the GABA receptor alpha 1. This is the zoomed image. We can clearly see the increased GABA receptor alpha 1 when the cells were transfected with the inhibitor of the MIR502 inhibitor. Same results is depicted on the basis of the fluorescence intensity. As compared to control, there is a significant decrease in the fluorescence intensity for this GABA receptor alpha 1 as compared to inhibitor. And this result has been also communicated for NRR. Then we wanted to check its effect on the GABA receptor alpha 1 more precisely by confocal microscopy. We have used same slides and observe at a high magnification by the confocal microscopy for the better imaging. And we find out in the control there is a GABA receptor alpha 1. We can see here in the merged image, this red color denotes the GABA receptor alpha 1 on its surface. When we transfect the cells with the agomer or the MIR502-3p overexpression, there is a reduction of this surface GABA receptor alpha 1. And we can clearly see there is an increase in the GABA receptor alpha 1 at the cell junction when the cells were transfected with the MIR502-3p inhibitor. This is the zoomed images for the inhibitor. We can clearly see. And same results were depicted on the basis of the fluorescence intensity. There is a severe decrease or significant decrease in the GABA receptor alpha 1 fluorescence intensity when the cells were transfected with MIR502-3p agomers. Then we have checked its functionality by the patch clamp analysis. So here we have used the HEK recombinant cell lines, recombinant HEK recombinant cell lines that produce the human GABA receptor alpha 1. And when in the parental cell, the GABA current was minus 20. And here in the scramble control, it is minus 24. And with the overexpression, it's remain almost same minus 24. But we can see when the cells were transfected with the MIR502-3p inhibitor, there is a increase in the GABA current up to 30. It's a minus 30, so current is in a negative, means it's increasing. Like minus 30, so GABA functioning has been increased when the cells were transfected with the MIR502-3p. Same results I have depicted here on the basis of the GABA current. So as compared to control, there is an increase in the GABA functionality when the cells were transfected with the MIR502 inhibitors. Then we wanted to check either it's the role of the MIR502-3p on the AD related proteins. So we have transfected the cells to the HT22 cells in the same way, and we have prepared the protein from the scramble control MIR502 agomir for the overexpression of microRNA and for the after inhibition after when the cells were transfected with the inhibitor of the microRNA. And we find out there is increase in the APP and tau level, but not much. You can see here by densitometric analysis, there is increase in the APP and tau, but this is not significant increase. But we can clearly say that, yes, there is an increase in the PSN1 and PSN2 level or protein level. There is a significant increase in the PSN1 and PSN2 when cells were transfected with the overexpression of the microRNA. And these PSN1 and PSN2 are the key proteins that play an important role in the Alzheimer's disease. So overall summary and conclusion. So we find out that the MIR502 targets GABA receptor alpha 1. It reduced the expression of the GABA receptor alpha 1, gamma 2, and B-glut. We have checked by the Western blot analysis. Then we find out that the overexpression of microRNA 502-3p inhibit the expression of GABA receptor alpha 1 mRNA and protein. Then we find out that the overexpression of microRNA 502-3p resulted in the cytotoxicity. It is cytotoxic to the cells. Then we find out overexpression of microRNA lowered the GABA receptor alpha 1 function at the cell junction by the immunofluorescence and the confocal microscopy. Then we find out that the suppression of a microRNA 502 increased the GABA function by the patch clamp analysis. Then we find out that the overexpression of MIR502 increased the expression of the key AD-related protein by the Western blot analysis. Thus, we can say that MIR502 could be a potential target to restore the GABAergic synapse function in AD. So I want to acknowledge my mentor and supervisor, Dr. Subodh Kumar at TTU HSC El Paso. I want to thank our chairperson, Dr. Rajkumar Lakshman Swamy. I want to thank our collaborator, Dr. Hemchandran Reddy, Dr. Lahiri, Dr. Rosel Swaldo, Dr. Bose, and my lab mates. I want to thank NIH Neurobiobank for providing AD and healthy postmortem brain sample. I want to thank NIH grant to my mentor, Dr. Subodh Kumar. Thank you. I want to thank my lab mates, Cheryl, Melissa, Danny, Alex, and my mentor, Dr. Subodh Kumar. Thank you. Thank you very much.
Thank you, Dr. Bhavinder, for your nice presentation and your research work. I think you are at the right place, and that is the things are here in India. The people, the scientists, also working on the same like Alzheimer's. There are many people in AIMS also, many people in Delhi and PGI also. the one group in Pulgay also they are working, like Dr. Bikash Mehdi group also working in this particular Alzheimer disease, and there are other people in India also. But the platform and the facilities are more at in US, like that you are also working in NIH with NIH also. You got the grant, and Subodh sir is also was also the part of PGI earlier.
Yeah, he has
He's a
obtained his
senior.
doctorate from the PGI Chandigarh also. Yeah.
Yes, sir. Yes. So I think most of the people they are there in the USA, and they are helping the US and NIH also, and getting more and more funding. And even Dr. I was in contact with the Professor Reddy sir even
Yes,
in
sir.
Reddy
Dr.
sir lab.
Hemchandra Reddy. He's the mentor of my mentor,
Yes, sir.
the super mentor.
I now I would like to like invite sir, Surendit sir, and anyone if you want to ask any question related to the research.
Dr. Prakash Halami. Dr. Prakash. Dr. Prakash Halami.
I'm requesting the audience, please mute their mic, or if they have any question, then please unmute. So you can get the benefit. This is the last day of the year. So we have very good speaker, and they have worked lot of work on Alzheimer's. So if anybody interested, they can just comment and ask about.
Yes, I request Dr. Preeti. Dr. Preeti, you can ask some question. Hello, Dr. Preeti.
Hello.
Yes, you can ask something.
Namaste, sir. It was very nice presentation, Dr. Bhupendra Sharma, sir.
Thank you. Thank you.
Sir, you have done a very wonderful presentation and very good research, sir. So,
Thank you.
like, I have some doubts regarding like you are working in particular with some of the enzymes.
We are working with the role of the microRNA. So our work is basically the role of MIR502-3p.
Yeah.
And its target is the GABA receptor alpha 1.
Okay.
We are working with the MicroRNA-502-3p. It targets GABA receptor alpha 1.
Okay.
GABA receptor alpha 1 is this subunit. That is the GABA A type receptor. So there is a reduction of this in the AD postmortem brain sample. We find out in AD patient there is a decrease in this receptor, GABA receptor alpha 1.
Okay, sir. Thank you very much, sir.
Dr. Tanvi, you have any question? Miss Tanvi?
No, sir.
Okay, no problem. And what about Kamaldeep Kaur? Kamaldeep Kaur? Dr. Prakash, you can ask some question.
Good evening, sir.
Good evening.
I have a little bit network issue here, but I think it is okay. I guess you will be able to hear me. No, actually, GABA, which is actually produced by probiotic bacteria or gut microbiota.
Pardon, sir? Pardon, please.
No, GABA basically produced by the gut microbiota, right?
So GABA is generally present on the cell membrane. It is the subunit. It's a transporter. Yeah.
Yeah. Now you want to tell that this particular mRNA modulates its synthesis or the transport.
So our work is like MIR502-3p. It reduces the GABA receptor alpha 1. Yeah, it reduces the receptor, but not the GABA. GABA is a neurotransmitter.
Yeah, exactly. So this microRNA can be used as a biomarker to
This microRNA.
identify the.
So in our previous observation, we find out that this there is a several microRNA, but this MIR502-3p, no one has
Yes.
published anything on this, and moreover, there is an increase in the MIR502-3p level with the increase of the AD Bragg stages. So in Bragg stage 2, 3, and Bragg stage 6, there is a significant increase in the microRNA level. So we can use this MIR502-3p as a AD biomarker.
And this is associated with anti-aging also?
Anti-aging? No.
Yeah. Somewhere you mentioned that this microRNA has a role in the aging, basically.
No.
In the aging.
No. It doesn't have, because we have used the AD postmortem brain sample from the healthy control, so they are age-based. So we have not find any increase in this microRNA with the aging, because we have used AD postmortem brain samples. Yeah, those unaffected control sample, they are age-matched.
Yes.
Those are the AD samples. So we find the decrease in the GABA receptor alpha 1 within the AD also, but no in the control.
Now, with reference to the from clinical side, how this information or microRNA can be used for treating the Alzheimer's disease patients?
So currently we have checked this on the cell level. Now we are moving to the mouse model. Our work is on the mouse model. So we are just, instead of the transfection, we are using transformation also, transduction also. We are using transduction with the help of the lentivirus. We are using this inside the mice models for the AD. So we have the different mice model. We just do the stereotactic surgery, and we are using this microRNA inside the cells.
Wonderful. Wonderful. Very good work. Thank you.
Thank you, sir. Thanks a lot.
Dr. Sharma, it
Yes,
was
sir.
an excellent talk. I really
Thank you,
enjoyed
sir.
your talk very much, and it
Thanks
has
a
got
lot.
a lot of applications.
Thank you.
Right? And
Yes, sir.
when you come back to India, will you continue the same work?
Yes, sir. Definitely, sir.
It is very much needed in India.
Yes, sir.
Because we
Definitely,
have
sir.
so many patients, especially the elderly people.
Yes, sir.
So your work will really help the population in India.
Yes, sir. And like our work is on a molecular basis. So like microRNA, they play essential role in in all type of disease. So yeah, we can use this microRNA as a cutting edge, innovative research in every other disease.
Yes.
In my previous PhD, I worked on the rheumatic heart disease, and we have find the role of microRNA also in RSP.
You should apply actively in medical colleges and also research institutes in India. Whenever you see an ad, you should apply from abroad.
Yes, sir.
Don't miss a chance, especially in CSIR and ICMR.
Yes, sir.
And congratulate you today, because your talk is the last talk of 2023.
Thank you, sir.
I congratulate you for that.
Thank you, sir.
Sunil,
thanks a lot. Yes.
Sunil, you can now take over.
Dr. Sunil, your mic is mute.
Yes. So, Bhupinderji, it's only done with the help of Professor Suranjit sir. He is my mentor also. He is my guide and everything. Because, without the help of Suranjit sir, we are nothing, because sir is supporting us, because he is, we can say, the superstar of Microbiologist Society of India. So, sir is helping actually the budding scientist. So all people are budding scientists. So I received your message, because even we conducted 5 days like faculty development recently, but at this moment, because it was difficult to add, but I requested, sir, that if you deliver your talk with the help of Microbiologist Society India, so that it will be better and a better platform, and many people, because we have many students right now, the most of the students not available due to the examination is going on. Some students are giving exams also, but the thing is that this talk will be live online on YouTube also, the MBSI YouTube channel. So it will be like watched with the thousands of like students will get the benefit out of your research. So I once again I want to congratulate you for your this research. It's a very big day for we people also, that we have an interaction with you. So all the best, Dr. Bhupinder, and
Thank you,
your
sir. It's only possible with you and also Dr. Sonjit Singh for this, providing me this opportunity and this
thank you
platform.
very much, and we
Thank you.
will send the sir will send you your this talk award letter, whatever the thanks letter, appreciation letter from MBSI. And we want in future that in 2014 you can also deliver your talks for like related to microbiology or related to your research. So our door is always open for you. The MBSI door is open for you also.
Thanks a
and we
lot.
want you that you can add your friends in our like MBSI. So we want like
surely.
contribution from your
Surely.
side. Yeah.
Yeah.
Thank you, sir. If you want to add something, then if I missed anything, I want to thank you all the team of MBSI.
Today, we actually arranged the talk of Dr. Bhupinder in just 6 hours time. In 6 hours time, I arranged your talk as the last talk this year. So that's why I congratulate you.
Thanks a lot, sir, and Happy New Year to all the members.
Thank you so much.
Happy New Year.
Thank you.
And thank you very much, sir. And thank you,
Yes,
Deshmukh Sirji, for providing this platform to us. Thank you very much, the president and Prakash sir. Thank you very much for joining us on this Sunday. And Happy New Year, sir, from Punjab, because we have lot of fog here, and we all are, inside. We are not going outside actually. We are going and we are coming back.
Yeah, in fact, I was in Delhi yesterday, so I know the situation there. Winter, yeah.
Yes, sir. Yes, sir. Thank you, sir. Thank you very much to all.
Thank you. Thank you very much. And see you next year, 2024.
Sure,
Yes,
sir.
sir.
Thank you. Thank you, everyone, and Happy
Bye-bye.
New Year to all of you. Thank you.
Thank you very much.
Thank you, sir.